The dengue virus by Araina, Vish, and Prapti. What is the dengue fever? The dengue fever is a mosquito-borne viral illness that is transmitted by infected mosquitoes to humans. There's 3 types of immunity that the body tries to fight the virus with. There's innate immunity, adaptive, and the secondary response.
The innate immune response to dengue virus begins immediately after transmission through the bite of an infected Aedes mosquito. As you can see right now, I'm currently drawing the mosquito that is used as a vector for the dengue fever. The virus is first deposited into the dermis of the skin, where it initially infects resident immune cells, including Langerhans cells, dermal dendritic cells, and macrophages. Viral entry occurs through the receptor-mediated endocytosis, which often involves receptors such as DC sign on dendritic cells. As you can see here, the mosquito is on the dermis of the skin, and the mosquito is injecting viral RNA into the dermis. There's 2 types of cells, again, that get infected first, which is dendritic cells and macrophages. So once the virus is officially injected inside the cell, as you can see here, this is a dendritic cell. It's like a flattened octopus-shaped cell. And there's also macrophages, which is more like a globular-shaped cell. They both have nucleuses, and they are the first types of cells to get infected with this mosquito. Once it's inside the cell, the dengue virus is an enveloped, positive-sense, single-stranded RNA virus. It fuses with the endosomal membrane, and its RNA genome is released into the cytoplasm and directly translated into a single polyprotein, which is cleaved into structural and non-structural proteins required for replication. As you can see right here, there is currently viral DNA, that is RNA, that is being injected into the cell. And in this next couple of drawings, I'm showing the polypeptide sequence that is currently being replicated inside the cell. Replication occurs on modified endoplasmic reticulum membranes, forming replication complexes. During this process, double-stranded RNA intermediates are produced, which acts as strong pathogen-associating molecular patterns. The mature virion is inside the cell right now. So replication, we're done with that. Now we're going to talk about recognition and how it recognizes which cells are infected and which are not. So there are currently a couple types of receptors. There's toll-like receptors such as TLR3 that detect double-stranded RNA, while TLR7 detects single-stranded RNA. In the cytoplasm, RIG-I and MDA5 recognize viral RNA and initiate antiviral signaling cascades. This is the basic structure of the RIG-1 sequence, and these are the receptors that activate adapter proteins such as MABS on the mitochondria and TRIF in endosomes, which trigger downstream signaling pathways that could lead to the activation of transcription factors, including interferon regulatory factors such as IRF3 and IRF7, as well as NF-kappa B. These transcription factors translocate into the nucleus and induce the expression of type 1 interferons, which is interferon alpha and interferon beta, as well as pro-inflammatory cytokines. Secreted interferons bind to the interferon receptors on neighboring cells, activating the JAK-STAT signaling pathway. This results in the formation of the ISGF3 transcription complex, which stimulates the expression of interferon-stimulated genes. These genes encode antiviral proteins such as protein kinase R, which inhibits translocation, and more that activates RNase L to degrade viral RNA, and MX proteins that interfere with viral replication. Simultaneously, cytokines such as TNF-alpha and more promote inflammation by increasing vascular permeability and upregulating adhesion molecules to cells. This allows circulating immune cells to exit the bloodstream and migrate into infected tissues. Chemokines recreate a gradient that directly directs neutrophils, monocytes, and NK cells to the infection sites through chemotaxis. NK cells play a critical role in controlling early infection, which they detect cells by sensing decreased expression of MHC class 1 molecules and increased expression of stress-induced ligands. NK cells induce apoptosis by releasing perforin, which forms pores and targets cell membranes, and granzymes, which activate capsases and trigger programmed cell death, which is apoptosis. There's also complement systems, which are activated through alternative and lectin pathways. These complement proteins such as C3b bind to viral particles, which enhance phagocytosis.
This is the primary response to the dengue virus, which is the body's first, slower, but highly specific reaction to a new pathogen. The dengue virus enters the body through a bite from an infected mosquito. Once it's inside, it begins infecting immune cells and spreading through the bloodstream. Dengue is a positive sense RNA virus, which means its RNA can be directly used by host cells to make viral proteins. Since this is the body's first exposure, it triggers the primary immune response, which takes time to fully develop, but it's very specific. Antigen-presenting cells like dendritic cells engulf the virus and break it down into smaller pieces. These pieces, called antigens, are displayed on MHC class II molecules to signal other immune cells. These antigen-presenting cells activate helper T cells in the lymph nodes. Once activated, helper T cells release cytokines that coordinate the rest of the adaptive immune response. In this diagram, the helper T cell is activated by the antigen-presenting cell. It then releases cytokines, which begin activating nearby B cells and signal them to respond to the infection. The activated B cells undergo clonal expansion, producing plasma cells and memory B cells. Plasma cells release antibodies, first IgM and then IgG, which bind to and neutralize the virus, while memory B cells remain for future protection. Antibodies help prevent the virus from infecting new cells, while cytotoxic T cells destroy already infected cells. This combined response helps eliminate the infection. Cytotoxic T cells recognize infected cells displaying viral antigens on MHC class I molecules and destroy them, limiting the spread of the virus. After the infection is cleared, memory B and T cells remain in the body. These allow for a faster and stronger response if the virus is encountered again.
In the first infection, the immune system successfully fought off dengue serotype 1, which was DenV1. Memory cells were formed and the infection was cleared. However, a second mosquito bite introduces DenV2, a completely different serotype of the dengue virus. While it's still dengue, the immune system has not encountered this exact serotype before, and so it doesn't really recognize it. DenV2 is now taken up by a dendritic cell at the edge of a lymph node. The dendritic cell displays DenV2 antigens on its surface and signals to the rest of the immune system that a new pathogen is present. This initiates the adaptive immune response. And this immune response rapidly triggers memory T cells and memory B cells and plasmablasts to generate from the first DenV1 infection. This is a characteristic of a secondary immune response because it's much more faster and much more robust than the primary response. However, these memory cells were originally trained against DenV1 antigens, and they only recognize DenV2 partially, but not with full specificity. Plasmablasts are rapidly generated, and their production is triggered by BLIMP1 transcription factors, which serve as the molecular switch, that convert B cells from a dividing cell into an antibody-secreting cell. Plasmablasts are characterized by a massive endoplasmic reticulum and high metabolic activity. The plasmablasts produce IgG antibodies, which are released into the bloodstream, and extracellular fluid. Inside the cell, antibodies are assembled on the rough ER, processed and modified through the Golgi apparatus, packaged into vesicles, and secreted outside of the cell into circulation. The bloodstream then becomes flooded with these antibodies shortly after infection begins. IgG antibodies that are present here carry out two primary functions. One is neutralization, in which the antibody binds to the virus and prevents it from entering host cells, and opsonization, in which the antibody coats the virus to flag it for destruction. Structurally, the antibody consists of a variable region that binds a specific epitope and a constant region, including the Fc region, which the immune cells recognize and bind to. The IgG antibodies produced against Dengue1 bind to Dengue2, but due to the serotype difference, the binding is non-neutralizing, which means the antibodies attach, but they can't actually block the infection. Macrophages express the Fc receptors on their surface, which are designed to bind antibody-coated pathogens and internalize them for destruction. When Dengue2 is coated in these non-neutralizing Dengue1 antibodies, macrophages recognize the Fc region and actively pull the virus inside. Rather than neutralizing the virus, the antibodies facilitate its entry into macrophages, and this is a mechanism called antibody-dependent enhancement, or ADE. Once inside the macrophage, Dengue2 releases its viral RNA and undergoes rapid replication. The infected macrophage releases large quantities of cytokines in an attempt to recruit immune support. However, the sheer volume of cytokines released overwhelms our normal regulatory mechanisms and instead produces a cytokine storm, which is when we have excessive dysregulated immune signaling, and this can cause systemic damage rather than actually targeting that pathogen that we need. The excess cytokines then act on the endothelial lining of our blood vessels. Under normal conditions, endothelial cells are held together by tight junctions that maintain that stability. However, the cytokine storm disrupts these junctions and causes plasma leakage out of that vasculature and into surrounding tissues. And this is what can cause severe complications that we will be discussing next. And this cytokine storm is further amplified by a phenomenon involving memory CD8 plus T cells. So these cells were originally primed against Dengue1. When they encounter Dengue2 antigens presented on MHC class 1 via T cell receptors, the molecular fit is suboptimal, or in other words, the fit isn't ideal. The T cells cannot mount an effective cytotoxic response and instead release large quantities of pro-inflammatory cytokines in a dysregulated attempt to try and fight the virus away. It uses prior immune memory against one serotype, but it actually just impairs the response to a related but a distinct serotype. For individuals who have never been infected with dengue before, or in other words, seronegative individuals, vaccination is not recommended. The vaccine acts like a first infection. It generates antibodies against all four serotypes. However, if a real dengue infection occurs afterwards, those antibodies cannot neutralize the new serotype effectively, triggering ADE, which means that it can actually increase the risk of severe disease in individuals that haven't had dengue before. But on the other hand, for individuals who have already had dengue, the vaccine kind of acts like a booster shot, so it reinforces and expands their existing immune memory across all four serotypes without the risk of ADE. The cytokine storm and the vascular damage covered earlier can lead to two severe clinical outcomes: dengue hemorrhagic fever and dengue shock syndrome, and both of these can cause leak plasma and bleeding across the body, as well as full circulatory collapse in extreme cases. The first approved dengue vaccine was Dengvaxia, licensed in 2015, and it covers all four serotypes and is approved for ages 6 to 16, and most importantly, it is only recommended for individuals with a confirmed prior dengue infection. But newer vaccines are working to address Dengvaxia's limitation. Qdenga does not require prior blood test. Butantan-DV has shown higher efficacy in clinical trials, and Dengue-All is expected to launch soon in India. And these vaccines aim to provide faster, safer, broader protection regardless of prior infection history. And together we must fight it. Thank you.